All right, last video on this regulation of gene expression section. We want to talk about how gene expression can be controlled after essentially the initiation of transcription. So this is not controlling the RNA polymerase's ability to interact with the promoter and initiate, right? But it comes after that. And these are going to be mechanisms that I'm going to tell you about that are controlled by structures in the messenger RNA. So the first one we want to talk about is a process called attenuation. And this actually involves translation and transcription actually interacting with each other. So translation, remember, can get started as soon as the region of the messenger RNA that interacts with the ribosome, in other words, the ribosome binding site and the start codon are exposed. And RNA polymerase can still be working downstream of that. And so the ribosome can actually interact with this, okay? So this sort of explains how it happens, but I want to actually show you this process here first, okay? So this, let me just make this a little smaller so you can see the whole thing. All right, so it says attenuation relies on coupled transcription translation. Okay, so let me see if this is actually a pretty nice video, so I recommend you watching it, but I'm going to try and sort of lay it out here, but I encourage you to watch this video because honestly, it'll do a better job than me. Okay, so here's what this looks like. This is in the trip operon. And so we have the promoter here and the operator for the trip repressor. And then these are the structural genes for the various enzymes for making tryptophan. But the key area we want to look at here is this leader region. This is actually just a short gene that makes a peptide whose job is not to do anything. It's essentially to participate in this attenuation regulation. Okay, so you have a start codon here, and then here's the most important codons. There are two tryptophan codons here. Then we have regions of this RNA that can interact with each other. Okay, so basically what this looks like here is that at high levels of tryptophan, when the ribosome gets to these two trip codons, it's just going to cruise right through because it's got plenty of tryptophanyl tRNA, charged tryptophanyl tRNA, to basically put these tryptophans in there and keep on going, right? Now, here's the regulatory logic here. If there's enough tryptophan around to be able to quickly do that, you really don't need to express the rest of these, okay? Because you've got enough tryptophan. If your tRNAs are charged up and ready to go, you've got enough tryptophan, all right? So if that happens, if those two codons can be quickly satisfied with their cognate amino acids, then we want to turn off the rest of this expression. However, if the ribosome has to hang out and wait and it gets stuck there because there's not enough charged tryptophan tRNAs, then the cell wants to express these. So if the ribosome gets stalled here, then we want to actually express the rest of these. So how does that happen? How does it happen that if the ribosome cruises through here, we don't want to make the rest of these, but the ribosome gets stuck here, we do want to, all right? Here's what it looks like. If the ribosome gets through this region, then the region 3 quickly, if it gets quickly through that region of the leader peptide coding region, then it gets into this region 2 here very fast, and there's not going to be any region 2 available to pair with anything else. But if it gets stuck here before it gets to region 2, then region 2 will pair with region 3. All right. Now, what are the implications of that? If region 3 pairs with region 4, this structure here looks an awful like a row-independent terminator. So you remember we talked about termination of transcription last week. That's after the RNA polymerase gets through the genes that it's of an operon or a single gene, whatever. So where it's supposed to stop, you have a terminator, right? So it's a stem loop structure followed by a run of A's in the template and U's in the synthesized messenger RNA. That ends up causing RNA polymerase to abandon this, right? So transcription stops if you make a terminator, right? So if region 3 and region 4 pair, you make a terminator, right? So region 3 and region 4 pair when the ribosome is cruising along fast enough to get to region 2 before it can pair with region 3. However, if the ribosome gets stuck here because there's not enough tryptophan around, then this region 2 here pairs with region 3 as soon as region 3 is made, and then we don't have a terminator because there's no run of U's after this, right? So if there's no terminator, the RNA polymerase just keeps on going and transcribes the rest of this operon, okay? So transcription continues if the ribosome gets stuck waiting here. Transcription stops if the ribosome doesn't get stuck. And the reason for that is the whole point of this whole operon is to make more of the tryptophan that would keep the ribosome from getting stuck at these trip codons, okay? So if you're starved for tryptophan and the ribosome gets stuck, you want to make more. So you need to transcribe this operon, okay? So why can this happen in bacterial cells? Why does attenuation work in bacteria but not in eukaryotes? You don't have this regulatory mechanism in eukaryotes. And the simple answer for that is because transcription and translation are physically, spatially separated in eukaryotes by the nuclear membrane, right? Because messenger RNA has to be exported out of the nucleus to get to the ribosome in order to be translated. This mechanism only works when they're physically in the same compartment, okay? Let's see. All right, so are there other mechanisms of RNA-based gene regulation? The answer is yes. One of probably the most common is antisense RNA, and that works by binding complementary sequences in messenger RNA with regulatory RNA molecules. So we call these small non-coding RNAs or sRNA, or yeah, we'll just call them that for now. Okay, so just an example here. I don't want to belabor this, but RPOS encodes a sigma subunit involved in stress responses. And so ordinarily, this is kind of weird, when there's no stress going on, the message is transcribed and it folds into a structure that blocks the start codon and the ribosome binding site. And this ends up being a substrate for a ribonuclease that inactivates this. And so you don't end up making the product of this messenger RNA. If there are various kinds of stress going on, you produce some small regulatory RNAs. One of them is called DSRA. DSRA will bind to this region of the upstream region of the RPOS transcript. And so the ribonuclease site is here, but basically this structure with DSRA bound actually leaves free ribosome binding site in the start codon for RPOS. So the presence of this regulatory RNA under stress conditions actually, binds to the RPOS transcript, but in such a way that it actually frees it up to be translated by the ribosome. A little bit counterintuitive, but this is just showing how that particular antisense RNA can actually activate translation of a message. Often, though, the antisense RNAs actually get in the way of translation, and so they are inhibitors of translation. So when a RNA molecule that blocks translation is made, it's a way of shutting down gene expression. So ribose switches are essentially structures in RNA that can control whether the message encoded in that transcript is effectively translated or not. All right, I'm actually going to stop there for tonight, and I'm going to end this, and we'll talk about this tomorrow. Okay.